Welcome to ASM Live, the internet talk show live from the 2013 American Society for Microbiology General Meeting in Denver, Colorado. Today we're going to be talking about the Merlot Microbiome. And so we have a couple of very interesting guests that will give us perspective on this. I'm your host today. I'm Stanley Malloy. I'm the Dean of the College of Sciences at San Diego State University and chair of the editorial board of Microbe magazine. I want to remind people in the audience that we'd love to have questions. If you have any questions, please come to the microphone to ask a question. And for those of you out in the internet world, you can send questions via Twitter to hashtag ASM 2013. So let's begin. Why don't you introduce yourself and tell us a little bit about how you got to where you are.
Okay, so I'm Iratxe Zarraonaindia. I'm from the Basque Country originally, and I came here to the US with a grant covered by the Basque government, so to be trained in metagenomics. And I'm currently working at Argonne National Laboratory in Chicago, and under the supervision of Jack Gilbert. And this is my story.
Very good.
Good. Thank you very much for inviting me, first of all, Stan. I think it's a pleasure to be here. My name is Francisco Diaz. I'm a food microbiologist and a professor at the University of Minnesota, Department of Food Science and Nutrition. And I've been in this job for the last 14 years. And before that, I went to school for my graduate work at Cornell University. I'm originally from Mexico. I came into this country 23 years ago. And I think I have different interests in my research on foodborne pathogens mostly, and also teach courses in food safety and food microbiology.
Well, why don't you tell us a little bit about your research and give us a perspective of what you're doing.
Yeah, in my group, our group is intended for, our focus is mostly applied food microbiology. We're targeting, one area is control of foodborne pathogens using applied methods. The other area is understanding ecology and the relationship of organisms with foods or with the environment of foods. I have to disclose, first of all, that even though I'm in this topic of metagenomics today, I haven't done research in metagenomics in my group. I just happen to be lucky that I put together the session as convener and also as a chair of the Division P, the Food Microbiology Division. And I'm learning a lot about metagenomics. It's a fascinating area. But in a related topic, some of the techniques we'll be using in my group recently is using transcriptomics to understand the relationship of pathogens like Salmonella, E. coli 157 with plant tissue, plant environment.
I actually think that's one of the exciting things about being a scientist is you change on a regular basis and have an opportunity to learn new things and shift. And very often you're driven by these external things. Somebody who gives a talk in your session or one of your students coming with something new. And
it's a fascinating field. It's just mind-boggling.
Absolutely. And how about you? Why don't you tell us about what you're doing?
Yeah, so my background is in population genetics, in fisheries population genetics. So this is a completely new field for me. So what we are trying to do is by using metagenomics approaches and characterize the microbial community associated with grapevines. So we have samples from vineyards in New York, in Long Island, and we are analyzing 4 Merlot varieties to trying to know which is the effect of this clone genotype in the microbial community, and then understand how the microbial community changes the productivity and the resistance for pathogens of the plants.
So are you looking at the community in the soil under the plant or also on the grape itself?
So we are characterizing all the tissues that we can. So we are analyzing soils, roots, leaves, flowers, and grapes, depending on the season. We have analyzed around 700 samples already, and we have quite interesting results where we see that the clone, there is a clone effect where each plant is selecting different microbes from the soil, and that these microbes are pop up to the leaves and grapes and flowers. So we are also interested in knowing which is the microbial community effect in the wine complexity. And this is hopefully something that we could say with the time.
Is there a reason why you picked Merlot?
When I came, the project was already started,
Okay.
and that was the variety that we were in contact with the people from the vineyards in Long Island, and in Long Island, the Merlot is like the star.
Okay.
But it's true that for me, it's a big thing, because in the Basque Country, we also have Merlot, and in France, Merlot is also very important. So we are also collaborating with people overseas, where we are, where they are sending us samples, and we are analyzing also these Merlot varieties from these vineyards.
Merlot sometimes gets a bad rap in the US.
Why is that?
I think it was all associated with one particular movie.
Okay.
I don't know which movie is that about.
You don't want to go there.
No. So when you do the metagenomics, you can see that there are these distinct communities associated with a particular variety. Have you been able to take and reconstitute a community with a different plant?
So this is something that we want to do in the future. So what we are trying to find out is to, for example, discover which are the microbes that are affecting in the productivity or in a disease resistance, and try to use these microbes to use them as probiotics, like to inoculate them in the soil or spray them in the plants, and to see or to better to improve the yield of other plants that don't have these microbes in their community.
You know, I think that the point you raise is really critical, and often I think that us wine consumers, when we think about the microbial communities associated with wine, we're focusing on the communities that alter the flavor, but diseases that influence the plants are really a critical factor in terms of availability of wine, and they tend to spread around the world very quickly.
Yeah, and we are also seeing these flavor metabolites in the roots. So what we are seeing is that roots are actually biosynthesizing a lot of metabolites, a lot of compounds that then are interesting for the wine, or are important for the flavors and taste of the wine. So with this, what we are seeing is that it's not enough with analyzing only the grape. We have to have a more integrative perspective of the plant and the microbes.
It's very interesting. This comes back to talking about foodborne disease, that some of the compounds associated with wine is believed to improve human health.
Yep.
And in fact has antimicrobial activities and potentially antiviral activities in some cases. So could you comment on that?
Yeah, no, I think, the Iratxe project is just one example of what metagenomics can do. I think it's a great application. But regarding foodborne pathogens, there's a number of researchers looking at what are for example, we have the problem with E. coli 157 or salmonella in vegetables like lettuce. There is some indication for whether the particular microflora that goes with each variety of lettuce may have a role in preventing contamination. So there is some relationship about those findings are being possible thanks to the advances on metagenomics. So that's one example. Another example you could find out is also at the pre-harvest level with animals. I guess there's going to be some of the other talks in the session we're going to be having this afternoon. We'll also look at the gut microflora and what is the role in one, for example, in preventing the colonization of livestock by some of the zoonotic pathogens. And you can also think about what are the role of our own microflora, why the problem we see, or the why some people get sick eating the same food and some people don't get sick. And I think there's, of course, the gastrointestinal microflora or the gastrointestinal function is we're still trying to understand the importance of that particular organ. But yeah, in summary, I guess what metagenomics can do is just incredible. We're just scratching the surface. And metagenomics, just I was looking in PubMed, for example. The first publication on metagenomics was in 2003. Last year, there were a total of 400 and over 400 papers that they had identified in just the name metagenomics. Those are the ones that you can just search with the metagenomics. So it could be more than that. And it continues to grow, as you can see.
Yeah, for me it's really interesting to see this because it's like I come from a completely different file and I will never expect that that the microbes are governing our world. Like there is microbes everywhere, their impact is everywhere. Like for me it's amazing. I wouldn't expect this before. Before coming here, I didn't know all this world, microbial world.
That's the message we want to send to the world.
Yeah, it's true, it's true.
No,
Yeah.
it really is amazing how metagenomics, I like to think of it like the discovery of the microscope. All of a sudden, you can see things that you were just oblivious to because you didn't have the tool.
Yeah.
And now that we have the tool of metagenomics, there are so many applications that it ties into population genetics, it ties into plant biology, it ties in very interesting aspects to human health, to you name it. There's so many applications.
Yep, yeah. I think it's a really promising field. What it will be able to deliver now and in the future too.
Yeah, but it's true that it's quite overwhelming sometimes because the amount of data that you receive from these sequencers is enormous. So we need a lot of bioinformaticians and a lot of development of tools to analyze all these sequences. Because it's true that like metagenomics is like the tip of, it's like the technology now in the tip of the world, but we need a lot of tools to overcome this massive sequencing data.
Yeah. Absolutely, we need more bioinformatics, and it's a lot of data. But a lot of times us biologists say that, and we sometimes forget that banks deal with a lot of money exchange, right, on a day-to-day basis. And if they're off by 1 penny, people will be pretty upset, right? And you know, the other place is astronomy. You think about the amount of data exchange associated with astronomy.
Oh yeah, yeah.
Big data is going to be our big challenge in the future,
Sure.
but metagenomics is only one of the aspects of that.
Sure.
That's
Yep,
important.
yep.
So I'd like to remind the audience, if you have any questions, please come up to the microphone. And those of you who are out in the internet space, if you have any questions, please send them to #ASM 2013. Yes?
It's a very interesting project. And I understand that you look at the impact of the clonal varieties on the microflora on the plants. Do you intend to also look at the impact of the microflora, the differing microflora on the plants, on the following fermentations? So will you look at fermentations also?
Yes, we are also interested in, when you say fermentation, you see the fermentation of the wine, no? So we are also interested in analyzing what is happening in this process. But to understand what is happening in this process, we also need to know first what is happening in the plant, because it's going to have a lot of implication on this step of the fermentation. So that's why we have to start first with the characterizing the system, and then we will go after the fermentation.
Thanks.
Thank you.
Another question?
Sheila Malloy from Nature Reviews Microbiology. I think I have a similar question, actually. I'm an enthusiastic consumer of wine, but I don't. So there are, as I understand it, a Merlot from California. It's slightly different to a Merlot from France, and I had always assumed that that was down to the cultivar of the grape. But is it possible from your work yet to say what the contribution of the microbiome is to that variation in flavour?
So actually, we have samples from Bordeaux, from France, that are cultivating the same variety that we have in our project. Also, so this variety is also grafted in the same rootstock, because another thing is the rootstock. Here, it's important also to highlight this. And so we are going to have a look on that in our next sequencing data that I have already received. So I will give you more data about that when I have all the data. But I think that, I mean, there is a lot of factors that could influence, that could have an influence on that, because we also see a geolocation effect, where we see that different vineyards, depending on where are they located, have also an impact in the microbial community. So this is another thing that could be affecting the flavors of the wine that you are seeing the differences in. Yeah, thank you.
So we can imagine someday the label of the wine will list all the microbes. No, I don't like that one.
Yeah, maybe you can fine-tune the flavor, I guess, and get it really consistent. If it...
Well, I think one place where this has already happened pre-metagenomics is really associated with the malolactic fermentation.
Oh, yeah, yeah, yeah.
And that discovery and the impact that had on consistency of wine production, right?
Yeah.
So, and that, of course, is tied so much to the types of microbes associated with the wine, both pre and during fermentation, and in fact, post-fermentation as well, right?
Sure.
So, and you're looking at each stage of that process.
Yeah.
So what you just said, I think, is very important, because you're going to be able to distinguish the microbes from the host genetics by using the same rootstock and the same varietal. So that will be really intriguing to see, in that case, exactly the influence of the microbes on the wine.
Yeah, like I was really interested in this clone effect, because the grapevine is in reality like two plants in one, where the roots belong to another species and the aerial part belongs to Vitis vinifera. So Vitis vinifera is like Merlot, Pinot Noir, all these varieties that we have. And Vitis vinifera is very susceptible for some pathogens, and that's why they are grafted in rootstock, because the rootstock is the one that gives the plant the resistance to some pathogens in soil. So I was really interested in the effect, or if there was an effect in these clone varieties, because they are all grafted in the same rootstock. So I wanted to know which were the interactions among above ground and below ground compartments, because they belong to different species.
Mm-hmm. Yeah.
Are there any other researchers doing similar kind of work, but in other wines? And let's say maybe in California or France?
There is a lot of people working in wine, but usually people work in fermentation. Also just in the grape.
Yeah.
And mainly most of the people analyzes the plant itself, not the microbes in the plant.
So you're pretty much being a pioneer in...
I mean, there is work done.
In this area. But yeah, one of the first ones.
Yeah, maybe this is a work where we have analyzed more than one factor. So we have analyzed different soil types, we have analyzed different seasons, different years, different clone varieties. So I guess this is the first more comprehensive study, maybe. And also, usually what people have been doing is use culture-dependent methods. So now with metagenomics, we can see a bigger reality of these environments. So I think this is also a point in this project.
Okay, good.
You know, a lot of wine growers that I've met, including people, I live in Southern California and interact with people in Baja California, and there's a fair wine industry there. Very, very concerned about the impact of global climate change on wine production. And that concern is largely associated with what's going to happen to the microbial communities in the soil as the temperature changes, as the amount of water changes, and all of these issues. So not only are you learning things that could help influence the quality of wine right now, but it may have a big impact on the industry in the future.
Yeah, sure. So together with metagenomics data and all that, we are also collecting a lot of metadata related with these samples, so temperature, latitude, longitude. So we are going to be able to correlate some of these microbial community shifts with the environment. So hopefully, if we continue with this project, we could be able to predict what will happen if there is suddenly a year where it's super dry or super wet. So we are going to be able to say these kind of things for the viticulturists.
Where's the funding come for this?
The funding comes from the Earth Microbiome project. So this is a multidisciplinary project where they are trying to analyze they are trying to characterize the microbial community all over the globe. So there are a lot of projects inside this big project, and using metagenomics, they are seeing things like the biogeography aspects of the microbes in different environments, and one thing that is really important from this project is that they are trying to standardize all the protocols, all the extraction and sequencing protocols, that all the projects could be compared to each other, because in microbiology, one of the big problems is that depending on the protocols that you use, you are going to obtain from the same environment different microbes. So in this way, standardizing all the protocols, we are going to be able to compare if what is being happening in one environment and in another has a relationship, a real relationship, or if it is biased by the process of analyzing it. So it's really important.
It's great to see that one of the national labs is focused on this area, because it is something of really profound national importance, not only because of wine, but because of impact on many other things, right?
Yep.
So how do you view metagenomics as changing your research and
well,
where
it's
you're going?
it's basically it's allowing us to be able to answer questions that before we never even imagined. I guess the power of the next generation sequencing allows us to get a lot of information in relatively little time. So and really address really complex systems, and actually being able to look at the systems approach, not only just a small part of the piece. With the techniques we used to have for many years, they restricted our ability to look at the system as a whole. So this metagenomics is changing our vision and our abilities to actually being able to better understand microbial communities.
So Francisco, give us an example. Something, so you gave a very big picture description.
Yeah.
But how about something a little more targeted?
Well, I guess, besides the Merlot example, which is a really good example, I guess another one that, let's say in my particular area of interest, which is foodborne pathogens, there is also the possibility of advances of metagenomics to be able to come up with the faster detection methods for pathogens. Whether you're able to get a lot of information really quickly about the presence of microorganisms in a food sample that may eventually lead to prevention of a foodborne outbreak. There will be also another presenter in the same session on some of the initiatives that the FDA is having on applying metagenomic techniques for this purpose of enhancing our ability to detect organisms. That in case of the food microbiology is really a challenge sometimes to be able to predict whether the food that we're going to be eating is may be safe or not. So that's where I see a lot of promise of applying metagenomics.
Now, one of the things that I think is interesting about metagenomics is when we first began to apply it, we'd look at a snapshot, one period of time. And so you don't see what's happening over time in those populations, any temporal or spatial changes. And you can imagine that there could be substantial changes in associated with the plant as the plant grew. Right?
Yep.
Yeah.
And
Yeah, sure. The developmental stage of the plant
Right.
is key also
Right.
for the microbes. Yeah.
So are you beginning to look at those types of things in addition to what's out there?
Yeah, I'm beginning with it. Beginning. Yeah, I have like so much data and so many things to look at that. Yeah, but yes, this is one of the things that we want to look at. Yes.
And it's very exciting to think about how this could be applied really broadly, and how you have so much data that those people out there, any students who are listening to the show, there really are great opportunities to move into this space, right?
Yeah.
So I'd like to remind the audience, please ask us any questions. If you have any, come to the microphone. And if you're out in the internet world, please send questions to #ASM2013. Another question.
Yes, I have one other question. There is a commercial available starter cultures like for malolactic fermentations. When you look at your wine fermentations, the metagenomics, did you have a chance to compare inoculated fermentations versus wild fermentations and
We
things like
hadn't
that?
arrived to this stage, so the fermentation samples are something that we are going to do in the future, in the next proximate future, but we hadn't analyzed yet the metagenomic data for the fermentation, so I cannot tell you more about that.
Thanks.
So many questions waiting to be asked. So another question from the audience.
Sorry. I just want to comment on the metabolite inference and fermentation, and also make a comment about the FDA and the generation of that. So one of the key things about this study is trying to identify the metabolites that the microbes in the plant could be generating that are then concentrated inside the grape. So it may not just be you activate or fermentate with a yeast, but it could be because the bacteria are killed off by the alcohol concentration in the fermentate. But the metabolites they generated beforehand can influence the wine quality and flavor. So that's what Iratxe is doing. But on the FDA, that FDA angle is very interesting. Being able to take metagenomics and use it in a clinical setting for prescribed quantitative estimation is incredibly complex. And very, you have to be very cautious of moving into that area to generate a quantitative estimation of where the microbes are coming from. There is currently a push towards understanding microbial loads in watersheds and in rivers around America, and individual water districts can opt out of the national mandate for their own monitoring and implement their own program for monitoring. So what do you see as being the potential to utilize this technology to potentially monitor pathogen loads in watersheds over that system?
Yeah, I think it's definitely, I haven't seen any work on this particular area of applying metagenomics to water, but I think it's a great application. I would say there would be a lot of possibilities. There's always been the concern about the fecal indicators, and it's been every year here at ASM we have a lot of presentations about the source tracking techniques, and I think it's metagenomics could potentially offer a really good viable solution, because there's always been the discussion about whether, of course, the classical fecal indicator is E. coli, but I think we need to go beyond that. And I think obviously the metagenomics offers the option of looking at organisms that may be present in intestine, but they're never culturable, and that's what now it's opened up the door. So I think it's a great, thank you for the question, because I think it's a great possibility for metagenomics.
Can I have one follow-up on that? Please. So that's exactly where I see the major benefit, is that, beaches around the world are closed on a regular basis because you go above that fecal coliform level. There's a set prescribed number of colony-forming units that's grown on a plate. And yet, if a bird population comes through and does what birds do when they land in an area, you see a significant increase in the colony-forming unit mandate for that. But none of those organisms are pathogenic to humans.
Yeah.
So you're closing beaches potentially unnecessarily.
Yes.
Now, the other thing is viruses. We have no real monitoring schema for viruses, and
Yeah, that's a
in
great
my
idea.
imagination, metagenomics could be a very powerful tool for identifying those pathogenic viruses.
Yeah, now we also have a talk on virus metagenomics, the virome talk. So come this afternoon.
In a previous edition of
yeah.
ASM Live, we talked about norovirus and the real serious problem with contamination of an outdoor park, and it's virtually impossible to clean up in real time. So tremendous economic losses associated with norovirus. So it's a case where a rapid detection method could be valuable. Well, you know, one of the things I think ties into thinking about the FDA requirements and the complexity of using metagenomics per se for analysis is once you've done a lot of metagenomics, it may give you a way of having a set of simple signatures that you can use in an easier, quicker test.
Yep,
for this.
and probably much more meaningful.
Right.
With just like the traditional look for coliforms and E. coli, and just you can have an array of different signatures and signals that will tell you much better information where the contamination came from, and if it is really a threat for the public.
Right. Well, this has really been an interesting conversation. I think we've gone from kind of the two sides of the issue, where metagenomics can really help understand plant health and the nuances of wine, and may someday allow people to manipulate it, to improve qualities of wines in ways that will allow us all to be drinking $70 bottles of wine for a much more reasonable. And secondly, I think the potential impact of metagenomics in being able to detect disease and improve health. So we'll have more time to sit around drinking wine instead of sitting on the toilet. With that, I'd like to thank the audience for joining us today at ASM Live, and we look forward to having you come back to another edition of ASM Live later. Thank you.
Thank you.
Extending the legacy of a classic reference, EcoSal Plus joins the ASM family of periodicals this June. This growing collection of continually published reviews reveals our ever-expanding understanding of the biology of E. coli, Salmonella, and the other Enterobacteriaceae. From cell architecture and growth to pathogenesis of infections to life in communities, EcoSal Plus is the single source of knowledge about these organisms and their use as model microbes. A distinguished editorial board, led by Editor-in-Chief James Caper, ensures that content serves the needs of the research community. Free trials available for institutions. Take a look at ecosalplus.asmblog.org.